If episode 1 in this series was about learning how to think about peptides, this episode is all about applying that thinking to the real world. Because once you understand that peptides are signals, that they're instructions and not supplements, the next question becomes unavoidable. What about the peptides people are actually taking right now? The ones marketed for healing, for injury recovery, for pain, for performance. These peptides are often talked about as shortcuts, ways to heal faster, recover better, or bypass the normal timelines of biology. Today, we're going to again slow things way down, not to dismiss these compounds and definitely not to promote them, but to ask a more disciplined question. What does the science actually support, and where does the uncertainty begin? So, welcome to Peptides 101, Part 2. And remember, knowledge is power. Hello, everyone, and welcome to the ReProgram podcast. I'm your host, Dr. George Murphy. Before we talk about specific peptides, let's re-anchor the framework from episode 1 of Peptide School. Remember, peptides are signals. They don't rebuild tissue directly. They don't provide energy or raw materials. They basically tell cells what to do. And when you introduce a signal into a complex biological system, remember the 3 negative outcomes that are always possible. Outcome number 1, nothing happens. The peptide is biologically ignored. You lose money, but not your health. Outcome number 2, something happens, but it's not what you intended. The signal hits the wrong target, or perhaps multiple targets. And of course, there's outcome number 3. The product isn't what it claims to be, or it isn't clean. There's contamination, maybe mislabeling, or sterility failure. And that's where consequences can become serious. So everything we talk about today will also fit into this framework. Okay, so why are healing and performance peptides so compelling? Well, mainly because they operate in a very emotionally charged space. Injury creates urgency. Chronic pain creates frustration, and slow recovery feels unacceptable, especially for otherwise healthy, motivated people. These can be people who are used to their bodies working well, people who train, people who take care of themselves, people who don't see themselves as patients. So when an injury lingers, it doesn't just hurt physically, it feels like a violation of identity. And in that space, people start looking for options that feel different from the usual pathways. They want something that feels more precise and less dangerous than, say, anabolic steroids, less invasive than surgery, faster than physical therapy, and perhaps more sophisticated than just wait and see. Peptides sound like that option. They sound targeted. They sound intelligent. They sound biological instead of brute force. And that's where social media enters the picture, because once you add transformation stories, recovery timelines, and confident testimonials, plausible biology can very quickly start to sound like proven medicine. Mechanisms become outcomes, animal data becomes assumed human data, and individual experiences get mistaken for general truth. That's not because people are reckless. It's because when you're injured or in pain, hope compresses timelines. And that's exactly where discipline matters the most, because biology doesn't speed up just because we want it to, and signals don't just become safe because they sound sophisticated. Okay, let's start with one of the most talked about healing peptides, BPC-157. Before we even get into the biology, I want to talk about the language that's always surrounding it. So online, BP-157 is often paired with something called TB-500, and together they're sometimes referred to as the Wolverine stack. And I really do get why that name sticks. I love Wolverine too. In the Marvel Universe, Wolverine wasn't special just because of the mythical Marvel metal fused to his skeleton. His real mutant power was his accelerated regenerative healing factor. So cut him and he heals, break him and he recovers, injure him and he comes back. So when people are injured, frustrated, or desperate to heal faster, that metaphor really does land. But metaphors shape expectations, and expectations can quietly outrun the evidence. So let's pause for a moment and talk about what BPC 157 actually is. So the name itself is already doing a lot of work. So BPC stands for Body Protection Compound, and the number 157 or 157 refers to the length of the peptide fragment. So that name didn't come from marketing, it came from early laboratory work. BPC 157 basically is a small fragment derived from a larger protein found in gastric juice, meaning that it was originally studied in the context of the gastrointestinal system. So early researchers were interested in it because the stomach has an unusual ability to protect and repair itself. Think about the very harsh conditions: acid, enzymes, constant turnover. You get the idea. So the original scientific question was fairly narrow. Could fragments of this gastric protein play a role in tissue protection or repair? So in animal models, mostly in rodents, BPC 157 did appear to influence several biological pathways that include things like blood vessel formation, inflammatory signaling, and cell survival pathways. So those findings are absolutely real. They're documented, and they're also biologically interesting. But here is the key distinction. What those studies primarily showed was signal modulation, not direct tissue regeneration. BPC 157 doesn't provide building blocks for healing. It doesn't become tendon, muscle, or ligaments. What it appears to do, at least in animals, is influence how cells behave during injury and repair. And that's an important difference because influencing a signal is not the same as controlling an outcome. But it's also why BPC 157 became so attractive beyond the gastrointestinal space. So once a molecule shows effects on blood vessels and inflammatory pathways, it's also very tempting to extrapolate those findings to tendon injuries, muscle tears, ligament damage, even nerve repair. But remember, extrapolation is not translation. So moving from animal data to human outcomes is where most promising biology either succeeds or quietly fails. And that's the part of the story that often gets skipped in online discussions. So, this big human data question, let's slow everything down and ask the question that actually matters. What human clinical data actually exists? And to be clear about what that means, human clinical data is not testimonials. It's not before and after photos. It's not podcasts, comments, or personal stories, no matter how compelling they sound. In science, human clinical data comes from well-designed studies where people are randomly assigned to treatment or control groups, where outcomes are measured objectively, where placebo effects are definitely accounted for, and where results can definitely be reproduced. So now when you apply that standard to BPC 157, the picture becomes very clear. There are no large, randomized, well-controlled human clinical trials showing that BPC 157 can accelerate tendon or ligament healing, can meaningfully shorten your recovery timeline, can reliably repair connective tissue, or produce anything resembling Wolverine-like regeneration in humans. So what does exist are small pilot studies, limited safety observations, and a large number of personal anecdotes. So these are not meaningless, but they are not evidence of efficacy. So anecdotes tell you what might have happened to one person. Clinical trials tell you what reliably happens across many people, and that distinction really matters. And this absence of strong human data isn't a neutral fact. Because if a compound could reliably accelerate things like tissue regeneration in humans, if it could consistently shorten recovery times for tendon, ligament, or muscle injuries, it would represent a major therapeutic breakthrough. That kind of effect would not be ignored. So pharmaceutical companies actively search for exactly this kind of biology, and they invest enormous resources into finding molecules that meaningfully change human healing outcomes. So if BPC 157, or any peptide for that matter, demonstrated that level of efficacy in people, it would already be the subject of large clinical trials and probably licensed or acquired by a pharmaceutical company and moving through formal regulatory pathways. And that's not speculation. That's how translational medicine works. And to be completely blunt, how this translation is funded and how it goes on to make these companies incredibly profitable. So when a compound remains outside that system for years, like BPC-157 has, despite widespread interest and, to be honest, widespread use, the most reasonable explanation is not suppression or oversight, and it's definitely not the conspiracy theories I've seen all over social media that propose that these companies or the government are actively preventing these compounds from getting approved and used by the populace. It's that the evidence required to justify that investment simply does not yet exist. And that doesn't mean curiosity is wrong. It means expectations should be proportional to the data. That's not an argument against exploration. It's an argument for humility in the face of incomplete evidence. Now, let's stay with this idea of healing peptides, but shift our focus more directly to something called TB-500. So TB-500 is often discussed alongside BPC-157, and as we mentioned earlier, the two together are sometimes called the Wolverine stack. If BPC-157 is framed online as a signal that helps tissues repair themselves, TB-500 is often framed as the amplifier, the peptide that makes healing happen faster everywhere. This framing is important because it reveals how people are thinking about this molecule. TB-500 is derived from fragments of something called thymosin beta-4, a protein involved in cell migration and wound healing pathways in experimental models. So in laboratory and animal studies, thymosin beta-4 does appear to influence how cells move towards sites of injury, how the cytoskeleton reorganizes itself during repair, how tissues respond to damage, and it's important to consider that this biology is real. And again, it is biologically plausible. But now we have to ask the same question we asked with BPC-157 and apply the very same standard. What human clinical data exists? And here, the answer is even more stark. There are no large, randomized, well-controlled human trials demonstrating that TB-500 accelerates tendon or ligament healing, improves recovery timelines, enhances performance, or safely promotes whole body repair in people. None. Zero. Zilch. So everything being claimed about TB-500 in humans is extrapolated, not translated. And this is where the idea of systemic healing becomes scientifically important for us to unpack. Because once a peptide circulates systemically, it doesn't know which tissue you injured. It doesn't know which tendon you care about. It definitely doesn't know where you intended the healing to happen. It interacts with biological pathways wherever those pathways are active. So cell migration, angiogenesis, which is the birth of new blood vessels, inflammatory signaling, and tissue remodeling are not niche processes. They are fundamental processes. They happen throughout the body all the time. So without controlled human trials, we don't know which tissues respond most strongly, whether some tissues are impacted more than others, how dose and duration of a particular peptide shift that balance, or whether effects resolve cleanly once this peptide is discontinued. So that uncertainty is not a technicality, it's the central issue. And this is where we can be more specific about off-target effects, what scientists are actually concerned about. So let's start with our friend BPC-157. In experimental models, BPC-157 does appear to influence pathways involved in things like we've discussed: new blood vessel formation, inflammatory signaling, cell survival, and stress responses. And importantly, these pathways are absolutely essential for healing. But they're also the same pathways involved in things that we do not want to accelerate. Blood vessel growth supports tissue repair, sure, but it also supports tumor growth. Signals that promote cell survival can help injured tissue, but they can also allow damaged or precancerous cells to persist. Altering inflammation can indeed reduce pain, but it also can interfere with immune surveillance, which is absolutely important. So to be clear, there is absolutely no evidence that BPC-157 causes cancer in humans. But there is also no human data proving that it does not, especially with repeated or long-term use. So in science, when a compound influences growth, vascularization, and the cell survival pathways, that uncertainty definitely matters. Now, let's return to TB-500. TB-500 influences processes like cell migration, cytoskeletal remodeling, tissue restructuring, and these processes are absolutely critical during things like wound healing. But they're also involved in things like abnormal tissue remodeling, fibrosis, and in certain contexts, cancer cell migration and metastasis. In other words, the spreading of cancer to other tissues and organs. So again, this does not mean that TB-500 causes cancer. It means that loss of specificity is a real concern. Cells don't change their behavior based on intent. They respond to signals. Without human trials, we don't know which cells respond most strongly, whether some responses persist longer than intended, or whether certain tissues are more sensitive than others. So off-target effects don't have to be dramatic to matter. They can be subtle. They can be delayed. They can appear only after repeated exposure. And this is why scientists are cautious about systemic signaling peptides. Once a signal is in circulation, it doesn't come with instructions about where to stop. And this is where the Wolverine metaphor finally breaks. Wolverine's healing factor was instantaneous. It worked in a perfectly targeted way. It was localized to his injury, and it was free of unintended consequences. Real biology doesn't offer that guarantee. BPC-157 and TB-500 are not mutant regeneration programs. They're signals introduced into a complex system that evolved to be tightly regulated. And without human clinical data, humility isn't pessimism. It's responsibility. Now, let's turn to another major category of peptides that comes up constantly in conversations about healing, performance, and recovery: growth hormone signaling peptides. So these peptides are often framed as smarter, more natural alternatives to growth hormone itself. So instead of injecting growth hormone directly, the idea is to stimulate your body to release its own more physiologically, more safely, and with fewer downsides. So I understand completely why this logic is appealing. Growth hormone plays a real role in tissue repair, metabolism, sleep, and recovery. But once again, logic is not the same thing as outcome. So let's talk about what growth hormone signaling peptides actually are. Unlike BPC-157 or TB-500, these peptides don't act on injured tissue directly. They work upstream. They interact with receptors in the hypothalamus and pituitary in your brain to influence growth hormone release, usually by mimicking or enhancing natural signaling pathways. The most common peptides people are using in this category include something called CJC-1295, sometimes with modifications, sometimes without, ipamorelin, sermorelin, growth hormone releasing peptides 2 and 6, and in some cases, tesamorelin, which is actually FDA approved, but only for a very specific medical indication. So online, these peptides are often stacked together, used in combination, and described as safer than growth hormone, more natural, easier to control. But again, this framing matters because all of these peptides ultimately act through the same hormonal axis. And with this in mind, we need to understand what things like growth hormone and something called IGF-1 actually do. So when studies show that peptides like CJC-1295 raise growth hormone and IGF-1, it's really important to understand what that really means. So growth hormone is produced by the pituitary gland, and it's released in pulses, especially during things like sleep, exercise, and fasting. So it acts as a master regulatory signal, and it influences how the body uses fuel, how tissues repair themselves, how muscle and fat are balanced, and how cells respond to stress. So one of growth hormone's most important downstream effects is stimulating the production of IGF-1, or insulin-like growth factor 1. So IGF-1 is where many of the long-term effects of growth hormone actually show up. So it directly promotes cell growth and proliferation, protein synthesis, tissue remodeling, bone density, and really important things like this, including muscle maintenance. In simple terms, growth hormone sets the signal, and IGF-1 helps carry it out. But here's the critical point. These systems, as we've discussed, are system-wide. When GH and IGF-1 rise, they don't just affect the injured tissue. They influence metabolism, growth, and cellular behavior throughout the entire body. Now, for some of these peptides, particularly CJC-1295, we do have controlled human studies that show increases in circulating growth hormone and IGF-1 levels. So that part is very real. But those studies primarily measured hormone levels and something called pharmacokinetics, or how the drug moves through the body over time. And generally, they just looked at short-term safety markers. They did not demonstrate faster tendon or ligament healing, improved injury recovery, enhanced performance, or long-term benefit in otherwise healthy people. So what we have is biomarker data, not outcome data. And raising a biomarker is not the same thing as improving your function. Okay, now let's talk concretely about what scientists worry about when people intentionally raise GH and IGF-1. First, unwanted growth signaling. Growth hormone and IGF-1, as we've discussed, promote cell growth and survival. Now, that's useful for repair, but it's not selective. In large human observational studies, higher circulating IGF-1 levels are also associated with higher risks of certain cancers, including breast and prostate cancer. And in conditions like acromegaly, where people have chronically elevated GH and IGF-1, cancer incidence is higher overall. So this does not mean these peptides cause cancer, but it does mean that increasing growth signals is not biologically neutral, especially over long-term horizons. Other things that are important to consider: growth hormone has a complex relationship with glucose regulation. And in some people, increasing GH signaling pushes towards insulin resistance, which you definitely do not want to have, because your body then has to work harder to maintain your normal blood sugar. And lastly, there are the classic GH-related side effects. When growth hormone and IGF-1 activity is pushed upward, well-described effects can include things like fluid retention, joint aches, swelling, numbness or tingling, carpal tunnel-like syndrome. And these effects aren't mysterious. They're consistent with what happens when growth signaling exceeds what the body is accustomed to. Now, there's one final point worth making. IGF-1 is not just a marker. It's an active growth factor. It promotes cell survival and reduces programmed cell death. Now, that's beneficial during development and controlled repair, but it can be problematic if it supports the survival or expansion of abnormal cells. And that's why endocrinology has always treated the GH-IGF-1 axis as something that requires tight regulation, careful dosing, and long-term outcome data. So the issue with growth hormone signaling peptides isn't that they're fake or that the biology is imaginary. It's that they act on a global hormonal system, not a local injury. And when you manipulate that system, you're changing how the entire body manages growth, metabolism, and repair. And specificity matters. That's why this category of peptides belongs in a very different conversation than simple supplementation, and why long-term human outcome data matters so much more here. Okay, so let's step back and put this all together. Across every category we've talked about today, BPC-157, TB-500, growth hormone signaling peptides, a consistent pattern emerges. The biology is often plausible. The mechanisms are often real, but the human outcome data is limited, incomplete, or entirely absent. And that distinction matters. Animal studies tell us what might be possible. Cellular studies tell us what could happen under controlled conditions. But human clinical trials are what tell us what actually happens across different bodies, different contexts, and over time. That's the gap people often fall into, because once a peptide shows a mechanism that sounds convincing, it becomes very tempting to assume the outcome. Mechanism turns into promise. Promise turns into certainty, and certainty turns into action, sometimes faster than the evidence can justify. That's not because people are careless. It's because when you're injured, in pain, or trying to optimize your body, hope compresses timelines. But biology doesn't just speed up because we want it to. And when people say, well, it worked for me, what they're often really saying is they haven't yet seen the downside. That's not a criticism. It's an observation about how complex systems behave. Science doesn't evaluate stories. It evaluates failure modes. It asks what happens when the signal hits the wrong tissue. What happens when it stays active too long? What happens when it interacts with systems we weren't targeting? And that's why throughout this episode, we've kept returning to the same framework. Outcome number 1, nothing happens. Outcome number 2, something happens, but not what you intended. And of course, outcome number 3, quality, purity, or sterility failures introduce real risk. So that framework isn't pessimism. It's how scientists stay honest. Now, if you're someone who wants to go deeper, if you want to see the actual data rather than just hear interpretations, I've listed the key peer-reviewed papers we discussed today in the show notes. I strongly encourage you to look at them for yourself, not to draw dramatic conclusions, but to see how careful, incremental, and cautious real science actually is. So, if there's one takeaway from today, it's this. Peptides are powerful because they are signals, and signals always deserve respect. Understanding what we know, and just as importantly, what we don't know, is how you replace hype with clarity. This is the ReProgram, and this series is about replacing hype with understanding. In the next episode, we're going to move into even more uncertain territory, because when peptides are marketed not just for healing or recovery, but for anti-aging, regeneration, and longevity, the stakes change completely. Those peptides don't just touch injuries, they touch time. They interact with aging biology, cancer risk, metabolism, and lifespan trade-offs, often with less human data than the compounds we discussed today. That's Peptides 101, Part 3, Aging, Longevity, and the Cost of Turning Up Growth Signals Later on in Life. I hope you enjoyed this discussion. If you did, please follow the podcast and join me on this journey of understanding and self-discovery as we navigate our ways through the fields of longevity, aging biology, regenerative medicine, and the emerging science of how and whether we can safely influence them.